Well, I want to thank everybody for taking the time today. I know these things, you get a lot of information fast, and it's a lot sitting there. So my goal is really not to tell you in detail about what I do, but I want to tell you a little bit about my research, give you a taste of what it'd be like to be in my lab, what my former students have done, and give you an idea of what I do. So my name is Biesiadecki. So I'm involved, my primary appointments in the Department of Physiology and Cell Biology, where I'm an associate professor. So I'm entering my 10th year here. So I'm also involved with the Davis Heart Lung Research Institute and the new Frick Center for Heart Failure and Arrhythmia that was just established. So if you can't tell, I'm interested in cardiovascular physiology. And I'm going to tell you about one of the, I just put this title up, this is really what's exciting about what's going on in the lab. But what I really want to focus on is what my lab is like. So I tend to keep a small lab. I like to be interactive with my people in my lab, with my students. My office is right adjacent to the lab, so our students and research assistants' offices are right outside, so we spend a lot of time talking in an informal basis. People come into the room all the time. We talk about science. So it's not just, my lab's not a place where you see me once a week. That's good for some people, other people it's not. But right now, my laboratory, I have 2 research assistants, so I have them listed here. It's Elizabeth Brundage and Brendan Agostina Boyle. So Elizabeth Brundage has been with me for almost the entire 9 years I was here. She was my first hire. She's the expert in biochemistry, keeps the lab running, keeps everything going. And I've listed 2 former biomedical sciences graduate students that have come through my lab. Benjamin Nixon graduated now about 4 years ago. He's doing his postdoc in Vanderbilt. And also Hussam Salih graduated 2 years ago. He's an M3 here now. Actually, I just saw him today. He's in his emergency medicine rotation today, but we're still finishing up a paper. So he'd been working on and off, but now he's back to clinics and pretty much done in the lab. So I've had 2 very successful students from the program here. Got a couple undergraduates, including Scott and Angel, working in the lab. And just sort of the makeup of the lab, I predict, will always be sort of small. So right now, like I said, 2 research assistants, 2 undergraduates. I'm looking to take a graduate student or 2, or a graduate student and a postdoc. So I'm very interested in building back up and getting more going. And what would that mean? Well, when you come into my laboratory, we're cardiac based, so I would expect that my students would take advanced cardiac physiology, so that's the 6101, 6102, it's a year-long course. It's actually advanced physiology, including cardiac. And then 8501 is our physiology class, that's an advanced cardiac physiology. And the other thing that's a little unique that we have in our departments, we have this thing called MCCP, and you would expect it to participate in that. And it's every 2 weeks we have this joint lab meeting with people that are interested in cardiovascular physiology. So it's 9 labs. We have lunch, and we all have our students present, to gain experience. If you're getting ready for a meeting, you present your poster there. The PIs present. Sometimes we bring people in. So this is a group of people, mostly in physiology and cell biology, all interested in cardiovascular research. So it gives you a chance to get involved, answer questions, in sort of a friendly environment, present your own work, and we do that every 2 weeks. The other thing I just came from is we just started a new thing with this, with the PIs only, sort of in heart failure and arrhythmia, so that doesn't directly involve students. But it sort of tells you about the collaborative environment that my lab works in with a lot of the people in physiology and cell biology, as well as Davis Heart Lung Research Institute, and I'll touch on that. So I'd expect my people would submit grants, AHA, NIH F grants for pre-doc. So I'm actually going tomorrow to review the F grants, so anybody that, whether you come into my lab or not, you want to submit for these, it's a good thing to do. Come see me. I can give you some pointers on how those work. There's some key things that are different than research grants, but that'd be one thing that I would expect you would learn. You would be expected to go to national meetings and present. So Hussam had 2 years ago when he left, he had a platform presentation at the Biophysical Society meeting, so that's a pretty big meeting, national meeting, about 6,000 people. And that was his deal, to give the presentation. So this is all part of the mentoring plan, that I really focus on the students, not only the research, but learning the research, being involved in collaborations, going out to meetings, learning to present, learning to write, getting involved in all these things. Dr. Raphael Fortenay will actually talk a little later, so I'm just going to touch on it about the T32 that I'm co-director of. So we really work very hard to make sure our students are developed, not only scientifically but professionally, and that comes down to my lab too. So we have a bunch of coursework thinking about, you know, just running a lab, picking jobs after. So to me, when I take a student, I ask that, as it says here, you know, a student's hardworking and a hard-playing student. So I take the responsibility, I kind of make a contract with my students that you're going to come in and you're going to work real hard for me, but on the other side, I take responsibility for you to make sure you succeed. So like I said, Husam's an M3, that's what he wanted to do. Benjamin Nixon's in Vanderbilt doing his postdoc, that's what he wanted to do. My side is to help you get through to where you want to be, your side's to work. And that's sort of the connection why I like to keep a small lab, keep in close touch with people. So I just want to touch really quickly on a couple slides. What do I do? Well, like I said, cardiovascular research. The heart is there to keep you awake by pumping blood, right? That's what the heart is. It's just basically a mechanical pump. So the heart contracting, the muscle contracting pumps the blood, right? And I'm interested in cardiovascular disease, so I just took this from the American Heart Association. So heart failure itself is a term used to describe a heart that cannot keep up with its workload. In other words, the heart can't pump enough oxygen, nutrients, and take away the disease. That's all. That's the definition of heart failure. So in the most simplistic sense, the way I look at this is if we have a person with heart failure, if we can improve their heart function, we can improve their outcome in heart failure. So that's, in the simplest sense, how I look at understanding things. So really the goal of my laboratory is to learn how to manipulate the heart's native regulatory signaling strategies that modulate cardiac contraction, and then we'll apply this as a therapy to help palliate the heart failure. So we don't think we can cause it solely by this, but we can slow down the development of heart failure, give people better life span. So we want to understand what happens in heart disease, why the heart doesn't contract well. We want to understand what happens physiologically, and then we want to basically trick the heart to do those things to improve the good things and stop the bad things, making the heart work better. And I'll tell you 2 slides on what I'm most interested in at this moment. So generally, most of you have been understand I'm very interested in post-translational modifications. If I say phosphorylation, you know, the kinase phosphatases signaling. Predominant phosphorylation is through serine threonine, and in the heart, those are the only ones known to directly affect cardiac contraction. So the beta-adrenergic fight response is phosphorylation of one of my favorite molecules at a serine residue. But about 5 to 10% of all the phosphorylation in your heart is actually tyrosine phosphorylation. It's very different from serine threonine. They're very separated. And we know that my favorite molecule, troponin I, is phosphorylated on a tyrosine residue. So we know that there are tyrosine kinases, including the Sarc family, in the heart. But nobody really knew what they did. So this was Husam Salih's project, who started this. So once it was identified, we went here, we used one of the methods where we take tissue from a mouse heart and we can exchange in our proteins that were phosphorylated. And this novel tyrosine phosphorylation that nobody knew what it did, we figured out what it did. It affects the heart function here. So now we know this is the first description of any tyrosine phosphorylation that can directly regulate cardiac contraction. So now I'm a little interested in tyrosine phosphorylation. It's always been kind of in serine threonine, and I'm sort of shifting gears a little. I'm getting interested. And where the lab's really going now is to understand this novel tyrosine phosphorylation that nobody knows. We know what it does for structure of the heart. It can affect hypertrophy and cellular signaling. But this is very interesting to me because we can't target well serine threonine phosphorylations. We tried this 20 years ago with beta agonists and we killed people. So the primary cause now, or primary treatment for heart failure, one of them will be a beta blocker. So we're affecting the serine threonine signaling. But serine threonine hits so many things. The tyrosine kinase is much more specific, and we now have a target that we know through this tyrosine kinase signaling can affect the heart's function and potentially improve that. So this is where the research in my lab is going first and foremost trying to understand how tyrosine kinase signaling directly affects heart contraction. So we made a few novel tools. There are no cell-permeable tyrosine kinase activators, so we made one in collaboration here with a chemist at OSU. And we made this, we can get it into living cardiac myocytes here. We can then measure their function and how they shorten. We can zap them, and we can measure how much they contract. And we just show here that if we take human myofibrils that we get from another collaborator, so every explanted heart that comes out of the Ohio State University, my other collaborator, Paul Jansen, gets, and we get some of that living tissue. We can treat it with this activator, and we can see that we can increase the tyrosine phosphorylation. So we have the tools now to start studying and understand what this means. So this is really where the project is at. And this is why I need a student to come in and pick up where Hussam left off and start carrying this project and these findings that he found. So some of the things that we do in the lab, we do a lot of biochemistry. So we're understanding how the different, the kinase affects and phosphorylates different proteins. We have made a transgenic mouse model of this that constitutively expresses this phosphorylation in its heart. And we've shown that the muscle from that animal, there's a different force and a different relaxation in that heart. So we do muscle level experiments from these animals. We also take these animals and we do hemodynamics in collaboration. So we do echocardiography in the living animal. We can recover it after. And then we do terminal hemodynamics measurements where we put a catheter into the heart. We can measure pressure volume simultaneously. And we know that these transgenic animals at baseline have a difference in their heart function. So I need somebody to come in now and figure out what happens in disease in this animal. So our disease model, one of them is that we collaborate with Mark Ziolo. He has a surgeon that goes in and does MIs on these mice, opens the chest, ties off the left anterior descending coronary artery, closes the mouse. We survive it. It has a heart attack. The question is, will our mice do better or worse with their different heart function in this MI model? So we're mimicking an MI in a mouse, and we can do all our muscle experiments. We can isolate cells. We can measure all their function. So we're gonna start modeling this disease in the mouse as well as in the human. So like I said, we get every explanted heart that comes out of OSU. We're working with Paul Jansen. He's isolating living human cardiac myocytes from people that do not have disease and people that have disease. And we're going to use our activators in those, and we're going to isolate those living cells. We're going to activate the kinases, and we're going to measure their shortening. We're going to measure the strength of that cell and see if it's better or worse. We're going to see if we can improve it in the disease and, in fact, if it hurts the normal. So the other area is that I'm really getting interested here in looking at other things. So this is just the contractile side. Nobody knows what this does to the excitation side or the channels and the action potential and the release of the calcium in the muscle. So we started exploring this, and through collaboration with Tom Hund and Peter Moeller, we're starting to look at these channel effects. And we already have a target channel that we know if we activate the tyrosine kinases, the phosphorylation on it changes and gives us a change in a living cardiac myocyte. So the future will be expanding this into other mechanisms of the excitation-contraction coupling, not only the contraction, but also the regulation of that. And we'll be doing this through adenovirus, different compounds. I told you about our activator in intact mouse human cells, our transgenic models. So we do a lot of biochemistry, a lot of Western blots. We do stuff in the muscle and cellular level with intact cardiac myocytes that we isolate from the hearts. And we do things in the animal. So I just want to wrap up a little, and I talked a little bit about the lab makeup. I talked about how, I see my lab and how I see the student fitting in and what I expect of a student and what I commit to the student. And I think the project you can see that I want to pick up and get going on. And then I just list some of the collaborators. One of the things we do is we do a lot of collaboration. So being in my lab is not just in my lab. I share a laboratory with Mark Ziolo. One of my rooms is shared with him where we isolate cardiac myocytes. We work with Jonathan Davis to do biochemistry. We work with Paul Jansen, Tom Hund. So these are all, for the electrophysiology. Dave Wupe was a chemist that helped us design these things. So it's a very integrative learning experience that you don't only learn what's in my lab, but you also collaborate with other labs leading to co-authored publications. So if there's any questions, if you need to find me, Department of Physiology and Cell Biology, Biesiadecki, you'll find me in the faculty list. Biesiadecki.1. There's no other Biesiadeckis here at OSU, so you don't have to worry about that. Can I entice anybody with a question about the heart? No? Everybody's falling asleep? Yes?
Do you guys have to see any physiological differences between hormones and
So what we do see is actually your heart relaxes quicker. So it doesn't cause disease because it's like, I don't like saying better because it's already, your heart's normal, it's good, right? So if you make it relax quicker, it's not better, but it's kind of primed. So when you have that hit of disease or you stress it, so another thing we do is we run mice on treadmills to stress them with exercise and stuff. What the prediction is they'll actually do better. So we've shown this through some other models and stuff we've done, but not with the tyrosine signaling. So we think they're going to do better in myocardial infarction, and that it'll potentially be a long-term target for treating people with MI and heart failure. All right? Everybody's excited? Well, thank you. Thanks for taking your time. I appreciate your time to listen.